and by our mentors. So I'd like to acknowledge Trevor, Torigay, for, just like guiding Logan through his research process and spending all this time at Iolani and working with them on the weekends and in the evenings. And now he's an honorary member of Iolani's wet lab. So thank you. And then we're really excited to see Logan Rubasch's talk, and he's put an immense amount of effort into this, and yeah, so thanks.
Good afternoon. My name is Logan Rubasch, and this summer I spent my summer doing research on how genetics affect ACL injuries. Before I start, I'd like to thank Mr. K for sponsoring the whole fellowship program. He gave me this opportunity to explore and kind of research and see what I like and see what's fun about it. I'd also like to thank my mentor, Trevor, who's guided me through this entire process, and he's taught me all that he knows, and it's been immensely helpful for me to kind of figure out all these complicated ideas and processes. And then I'd also like to thank Dr. Chan for being there to keep me on track and just make sure that I'm getting all my deadlines and all that. Yeah. So, shall we begin? ACL injuries are the most common knee injury in America. Over 250,000 cases happen annually, and two of the main offsprings of that problem is that there's a high re-injury rate, and osteoarthritis within 15 years of surgery is also really important aspect of why these ACL injuries affect us so much, especially financially. This diagram on the left just shows how many high school ACL injury rates occur each year, and like girls soccer and girls basketball both have really high ACL injury rates. On the right there is just a diagram of where the ACL is in the knee joint and how it kind of goes across the knee, and that's where a lot of pressure is being put on when we're jumping or running, and that's kind of why it's so highly injured or so commonly injured. So Trevor brought me onto this project that's kind of looking at a bunch of different factors that they were looking to see if they could correlate the likelihood for ACL injury to all these factors. So biomechanics are just how our legs move and how we're planting when we jump, and maybe that has an effect on our likelihood for injury. Physiology, so we're looking at the blood and how different proteins in the blood and the amount of those proteins might affect or might show fatigue or muscle strength that might lead to our ACL being more likely to tear because it's not properly supported. Cellular biology just looks at the physical makeup of the ACL and tissues around it that are looking to see if the ACL is different from the tissues that are being used to graft it and why the re-injury rate might be so high, as well as just what's in the ACL that makes it maybe less or more lenient to injury versus other knee tendons and ligaments that are being injured less often. And then genetics and gene expression, we're kind of seeing what genes affect our ACLs and what role that might play. And then gene expression is what we were looking at with a gene called FOXO3, which I'll get into. So we were relating genetics to the propensity for ACL injury, and for that we were looking at kind of what the ACL is made of and what this one gene FOXO3 what the effect of that gene is on the ACL and the composition of the ACL. So collagen is the majority of what the ACL actually is made of. So collagen is these little fibers that they expand and contract, and they can elongate to a certain point. But once they go past that point, that's where they snap, and that's where you see ACL injuries and ACL tears. And then this gene FOXO3 has been is part of all these different processes in the cell, but the main one that we were looking at was the telomere length and how it's related to cell degradation. And if one there's like the TT, GT, and GG, and if people who carry either the GT or the GG gene have a significantly higher chance of living a lot longer. And that's because the telomere lengths on each of our DNA strands are lengthened, and so we're looking to see if this increase in the telomere length or this increase in cell longevity might lead to an increase or decrease in the cell longevity of the ACL cells. And so we're kind of trying to correlate FOXO3 to the ACL and how it's decomposing as age goes on and different things like that. So another thing besides age that we were looking at was the graft tissues used in the ACL reconstruction surgeries. And for that, there are about 3 or 4 main ones: the patellar tendon, the semitendinosus tendon, and the PCL, the posterior cruciate ligament. And so those are kind of common graft tissues, and we're looking at the FOXO3 expression in those tissues as well. And then that's just on the left, that's kind of a diagram of all the different structures in the knee. And then on the right is just a picture of post-surgery, how different or how inflamed it is after the surgery and how much swelling there is afterwards. So my question was, how does FOXO3 expression in the ACL change as we age? We were looking at 2 cadavers that were about 19 years apart, and we're trying to see what effect the age difference had on the amount of FOXO3 expressed. For sample collection, I wasn't personally allowed to take part in most of this because it was mainly done at JABSOM, the UH's School of Medicine, and I didn't have the clearance to actually be working on the cadavers and on the samples. So Trevor and his colleagues were the ones who extracted the tissue samples from cadavers, and that was in the anatomy room, and then they were extracting the RNA from those tissue samples. And since we were looking at gene expression, we used RNA because if DNA is expressed in all the cells in our body, the RNA is only the ones that are being coded for, and that's what's actually coding for the proteins. So that's kind of why we were looking at RNA. And one problem with the RNA was that once you extract it, it's really hard to keep it from decomposing. So we turned it into cDNA, which is complementary DNA, a much more stable copy from which we can quantify the number of RNA strands that were actually in a sample. As you can see on the left, that's the transcription that occurs, and that's what is happening in our cells, and that's why we're measuring the RNA. And then on the right is the quantitative polymerase chain reaction process that kind of goes on, and that's what we were using to quantify our data. So for the qPCR, it's just the cDNA, we take the strands of cDNA, and then this machine splits the DNA by denaturing the strands, and then attaches a marker that glows a little bit. And so each time they split it and then attach the marker, it gets a little bit brighter. And so you have this sample that's increasing in brightness as it goes on. And then eventually it'll hit this threshold where the computer will, in my next slide, so the computer will actually measure this threshold. I don't know if it's kind of hard to see, but there's a green line there. That once it hits that threshold, the computer measures this time that it took to hit that threshold, to hit that brightness. And from there, we can kind of see this one sample took less time to make X amount of variation or X amount of replications. And so from there, we measure and compare our different gene expressions. So that's our cycle threshold value, is where it kind of surpasses that green line. And so to the left, if it's less cycles, then that means there was more of that gene being expressed. And if it's more to the right, then that means that there's less of it being expressed. Our delta CT, or the difference in our CT values, we were measuring FOXO3 expression against this gene called HPRT, which is kind of ubiquitously expressed in our entire body. So we can kind of use it as a baseline to say if FOXO was measured this much more or this much less than the HPRT gene, that's kind of what we use to normalize all our results. And then from there, we could compare the FOXO expression versus the HPRT expression in one sample versus another sample. And so our measurements were always either tissue to tissue within the same cadaver, so it would be the ACL compared to the ST, or the ACL compared to the PCL, or something like that. or the cadavers, where we would use cadaver 2's ACL sample and compare it to cadaver 7's ACL sample. And for that, that's kind of how we're quantifying our data and figuring out what was statistically similar and what was statistically different. So how is FOXO expressed in the ACL and its graft tissues, and how does this change with age? So we found that there were significant differences in the FOXO expression between different tissues and ages. This bar on the left is showing the cadaver 2's ACL. We took 2 samples of the ACL from the same cadaver, and that was cadaver 7, and so we kind of just compared those, and as you can see, those are really significantly, they're the same pretty much. And so the other 2 samples the 2 times we compared samples and they were similar was with the cadaver 7's ACL and cadaver 7's semitendinosus tendon, as well as cadaver 2's semitendinosus tendon compared to cadaver 7's semitendinosus tendon. And the numbers might be a little bit confusing because those were like the cadavers that were in the anatomy lab, but it's not necessarily like cadaver 1 and cadaver 2. So what was different was the cadaver 2's ACL compared to its semitendinosus tendon and its ACL compared to the cadaver 7's ACL. From that, we could kind of use this fold change to quantify what age is doing to the gene expression of FOXO. Even though we only used 2 cadaver samples, we kind of got this fold change where we use the logarithmic function to determine if it's positive. If our numbers were positive, then that would basically mean that FOXO was increasing with age, and if it was negative, then it was decreasing with age. And so what we found is that both the times we used the 2 ACL samples in cadaver 2, and we measured both of those to the ACL sample in cadaver 7, and found that it's decreasing as age increases. So what that means for age-based injury prevention, basically the implication of that would be that hopefully someday we'll be able to take a patient's genotype and say, look, you have this FOXO gene and you are this X age, and so we might be able to say that the collagen or the internal processes that are affected by the FOXO within the ACL are kind of deteriorating because the FOXO is being expressed less and less as they get older. The other thing is grafting options and how we're looking to see if the FOXO expression between these different graft tissues are different, and so they affect the collagen content and the different actual physical makeup of these tissues. And so that will make it easier to tell maybe this tissue is better to graft than this one because the re-injury rate might be higher with, say, semitendinosus tendons or PCLs. And so yeah, that was my research with gene expressions and genetics, but I also did a lot of work with blood physiology and relating that to strength this summer. So my whole month of June was actually spent working on the blood physiology of creatine kinase and actin-3, which are 2 proteins that are commonly related to strength. And so I got to learn a ton about that process and how using Western blots to just kind of show what's going on and how much protein is actually in certain samples of different people's blood, and we're going to relate that to the strength and fatigue. And that's kind of another part of the entire project that I got to work on. And so I learned just as much about that as I did about the ACL gene expressions. Thank you. Yeah, I would like to thank Mr. K for just putting this on. I'd like to thank Trevor for being there and teaching me all of this stuff, and I would not understand it if it weren't for him. I'd like to thank Dr. Chan for just, again, like keeping me on task because she was worried about this since day one. And then I worked with Lexi Masca and Maya Wu over the summer, and we worked a lot, and it was helpful to have other people just as confused as I was. Yeah. Any questions? start early. I think I like was past the deadline when I signed up. I don't know. I forget, but it was really bad. But it is a great opportunity, and like it's really fun to figure out what you might want to do, and especially since this is such an interesting topic, it's so worth it, and it paid off in dividends for me.
Will you take this out to science fair?
I'm not sure. I'll see what time permits. Yeah.
Just curious how you landed on this particular research.
I mean, it seemed like Trevor was looking for people from Iolani, and he was looking to kind of get in and work in the wet lab and kind of have a part in this whole process and have Iolani be a part in this whole process. So I think he contacted Dr. Chan and she just kind of told me, hey, if you're interested in looking at ACL injuries, he's our guy. So yeah. All right.